So today's speaker is Professor Jamie Morris. He did his PhD at Stony Brook University, went for a postdoc at Duke and then to Yale. And at some point he intersected with Kevin Pelfrey. I don't know whether it was at Duke or Yale or both. And then he came to UVA. Jamie has used multiple modalities, MRI, fMRI, EEG, event-related evoked potentials to understand how our brains understand what's on other people's mind, what we neurologists call theory of mind or TOM. But Jamie Morris, welcome.
Thank you. Happy to give this talk. It's a different talk than I normally give. I learned to do imaging work with Kevin. Kevin taught me how to do imaging work in the context of learning to do it with individuals with autism. And then since then, I've really gone on and done a lot of work in social behavior. And that's where most of my interests are with neuroimaging stuff. We do a lot of work though in children as well. And over time, as I've used functional neuroimaging, I've found a lot of sort of useful ways of thinking about it methodologically. And this project really came out of a lot of just sort of, chance, so to speak, just some nice collaborations that came to be, and they're still sort of building on top of one another. So it's been kind of fun. The way that I came about this is I started with Tim Salthouse, and Tim Salthouse, for those who don't know, was a professor in the psychology department at UVA for many years. He came to UVA from Georgia Tech somewhere around 2000, and he started the Virginia Cognitive Aging Project, known as VCAP. And VCAP was unique in that Tim really rigorously tested these individuals over as many chances as he could get, as many summers as he could get, really. And he was looking at aging from people that were 18 to 100, essentially. He was looking at aging all across the lifespan. He was taking into account a lot of unique factors of testing, a lot of things that will influence results. He was doing a lot of the mechanistic work that one would have to do to understand how you relate people's sort of scores on these types of tests to their true sort of nature, how much they're really declining cognitively. This is just some stats I had from a while ago. Tim retired 2 summers ago now. And since 2001, there have been 5,000 adults participating. Each one of the participation sessions were really long. In fact, they would have 3 sessions that people would come in over the summer, and there'd be 2 hours each with alternate versions of different cognitive capabilities of tests that you'd probably hear of a lot in terms of neuropsych testing, cognitive aging testing. And then he would make associations both from a cross-sectional standpoint, so looking at various age groups across time with different subjects in it, or longitudinally, so being able to follow the same participants over time and watching as they change. Individuals, he tried to test them every 2 years. He's very rigorous about this and didn't want to sort of test too frequently, because what happens is you actually see some testing effects where people get better in short periods of time. Just to give you a little flavor of some of the things that we have information on some of these participants, they have tests of episodic memory, reasoning, spatial visualization, perceptual speed abilities, questionnaires. They're extensively phenotyped across a number of characteristics, physical activity, depression inventory, anxiety, satisfaction with life, a couple of social measures here in terms of social network, various environmental factors as well. So these people were well tested, and they were well tested over time. So I just put these out here, not to belabor any one of them, but just to kind of give you a flavor for what it was like to be in this group, this VCAP group. Now, what's it like to age over time? Well, I guess the good news and the bad news is that I think everyone on this call is declining in some way cognitively. In fact, we start in our 20s, we start to decline in a lot of cognitive skills, including memory, reasoning, spatial visualization, and speed. And you can see this in a cross-sectional way. This is data from cross-sectional aging. Tim Salthouse's paper in 2009 went over this. He was making some larger points about how skills over time diminish. They diminish at varying rates depending on what skill you're talking about. And there's some differences with longitudinal versus cross-sectional that, again, were the focus of a lot of Tim's research. It's not important here. I just want to make the point that when you look at aging as something going on from 20 till 60, when you look at something like that long of a period of time, this is what it looks like. And this is going to be a relevant point that I'm going to try to make later when I'm going to tell you that this is one of the problems that we have when really understanding aging, is because we have to understand it at such a large scale. A 20-year-old and a 60-year-old, you know, any one of us can tell they're different. They're different in any number of ways, including on these cognitive dimensions. And when you look at it in this long scale, some of the things that we take as dogma become less sort of interesting to what's really happening at the really micro level that's really potentially a sign for clinically relevant details or some factor in terms of our understanding of how the brain changes over time. VCAP imaging, so how this came about. In 2016, Tim had approached me about doing some imaging on a small sample of individuals from VCAP. He was somewhat motivated by this because though he got a perfect score on his last submission for his R01 for VCAP, the one criticism in there was that Tim never had any biological measures. And with a lot of the work at the NIA at the time, the National Institute of Aging, and a lot of these other big longitudinal groups, his sample sort of stood out as a sample that had a lot of behavioral data, but no brain data. And that was very different for a lot of these other longitudinal samples that had been collected. So he wanted to get his hands on at least just a subset of individuals from the VCAP that we could recruit and bring in and do some imaging. He was specifically interested in some simple measures that had been reported in the literature in terms of gray matter degradation and some functional connectivity and some various networks. A couple of networks at the time were sort of the ones that people like to report on and discuss. In 2017, Morgan Lynch became the lab coordinator for this project. She had worked with Tim before, and she did just a tremendous job of collecting these samples. So we collected right after the scanner upgrade. We were kind of pushed off into the middle of 2017 because we waited for the scanner upgrade at the time over at the Snyder building. And we started in the summer and we ended in the fall, and we collected 90 some people, or 99 people. And we had participants that were between 58 and 81 years old. So we were aiming for between 60 and 80. Tim included a couple of people that were just a little bit below 60. We had 99 total people. The mean age was 68. They all had at least 2 testing sessions in the past. So these were people that there was some information on going in. Tim had actually done a little bit of work to select this sample. He had tried to choose some people going in that were aging gracefully and some others that were showing sharper measures of decline between these two last sessions that they had had. As it turned out, when he tested them again, as a statistics teacher, I have to tell you a little bit about regression towards the mean. When you select samples off of large characteristics like that, they weren't declining as much as he had hoped. So it was sort of being brought back to the middle. So it's not a random sample of VCAP participants, but it's not completely non-random either. I'm not going to make any inferences about who's who in there, just to say that we do have a mixture of people that were aging pretty well and some others that maybe weren't aging as well. Okay, so that was that. So that happened in 2016, or 2016 is when we conceived it. 2017 is when we collected it. We did some functional connectivity. We did the stuff that Tim wanted to do. I'll get back to that in a little bit. We didn't find a ton out of that. Tim's hypotheses were very, very narrow. You know, we found some confirmatory things, but nothing to jump up and down about. I didn't do much with the data past that point. I worked a little with my student, Megan Puglia, who was doing a fMRI experiment with them. And we did a little bit with that as well, but it kind of sat there for a bit as we went through. And then another call came out from the Brain Institute again about grant opportunities. And this was a sort of a fortuitous chance. I was working with Jessica Connolly, who was working with group here in psychology, Joe Allen's group, studying a longitudinal sample of people that Joe had followed from adolescents who are now into their 30s. And they were doing something called epigenetic aging. So they were doing a project looking at using people's whole genome scan information to create an epigenetic age, which relates to all different measures of health variability. And because we had talked a lot about this in our joint lab meeting, I thought this would be perfect because we had collected blood on every one of the VCAP participants. We could actually get some epigenetic aging on these guys, and then we can really start to try to understand some of these functional networks, maybe with a more precise measure in this case. So thanks to the generosity of the Brain Institute, we were able to pay for some whole genome scanning of this blood that we had banked, it's DNA that we had isolated. And then working with Jess, and I'll point out each one of these students as we go by, but Josh Danoff, who works with Jess in the lab, did all the work in terms of the bioinformatics with the data coming off of the genome. Amalia McDonald has been leading the charge with working with our functional connectivity analysis, which I'll point out. Sam Bridley's been working with Amalia. There's some data that she has just generated that I'll show today. And Ben Newman works with Jason Druskull, and he does white matter microstructure. So I'm going to talk a little bit about some structural imaging today as well. So a bunch of different pieces here, which I think are coming together nicely. Our goals were to use an epigenetic biomarker of age to predict connectivity differences in our elderly sample. That was the large goal. It's very descriptive to start with. It didn't have a lot of specific predictions about specific networks, but I will talk a little bit about some networks that are emerging and where that sits in terms of the literature and how we think about them now. And ultimately, we want to relate some of this to a lot of this cognitive data we have on them. I won't have as much to say about that today because I've been spending all my time thinking about the brain and the network data and how that works. And so we're going to take that apart and hopefully then be able to relate it back to thinking about some of the cognitive data and what some of those predictors might have told us about these changes in these networks. Okay, so DNA methylation and epigenetic age. So years ago, the National Institute of Health, recognizing that chronological age is a great measure and it's a lousy measure, right? So every old adage you say age is just a number, people age differently. Some 60-year-olds look great, other 60-year-olds look like they're close to death. Whatever you want to look at it as, chronological age is not terrific, but at the same time, overall, as I showed you in that one scale, if you look at it from 20 to 60, it is great. It is a great predictor. It's a great predictor of change. Things change as you age. That's something that you could find from every developmental study ever. Things change as you age. But is there a more precise way of looking at maturation biologically? And they asked researchers, we need better biomarkers of the aging physiological systems, our brain, things that are going to relate to cardiovascular disease, cancer, all risk mortality. We need better markers of advanced aging where we can see it. So people went in a search, so to speak, for sort of things that change with age and whether or not they are useful biomarkers in this way. One of the places that came up to be the most fruitful for this search is in the methylome across the genome. So DNA methylation is an epigenetic process. There are 28 million CPG sites along the genome, and most of them, the majority of them, not most of them, but the majority of them are 100% methylated. So in this case, what happens is you have a methyl group that attaches to the cytosine at the CPG site. So where you have this C and a G, you have this methyl group that attaches there. Okay, and we know that this is highly tissue specific. We know that this is how we have differentiation of different tissues. This is how we have different systems. This is how we have different sort of phenotypic variability in a lot of different domains. We can take all these cells from one tissue, for instance, in this case from blood, even though blood's made up of different cellular composition, we can worry about that a little bit later. We could take a bunch of cells and we can quantify the percentage of alleles that are methylated from the cells. So how many cells are showing this level of methylation. And then we could express this as a percentage, 0 to 100%. As I said, a lot of places in the genome, it's just 100% methylation. Now, when you talk about this in terms of epigenetics, a lot of times people talk about the impact of methylation on gene silencing. And that's a nice canonical way of thinking about it. So something that prevents transcription in this way. When we're talking about it today, I'm just going to be talking about one property of methylation that's interesting for what we're going to discuss, which is that methylation changes with age. Methylation across the genome changes with age. And this is for a lot of reasons. And the construction of clock doesn't have anything to say about the mechanisms by which that happens. There's environmental reasons why methylation could change with age on the genome. There's just regular biological processes. There's cell division issues that can go on. There's, for any part of the genome, for any cell, there could be a number of reasons why methylation changes with age. But the point being that it does have this nice movement with age across the genome. So knowing this, what researchers did was they said, okay, so if this changes with age, is there a way we can take a measure of methylation across the genome, find relevant CPG sites, which ones are most diagnostic of this change, which ones are the ones that line up most with the change chronologically. So the first kinds of clocks that they made were essentially which parts of the genome track with chronological age. That way we can come up with an epigenetic clock. And that one will fit really nicely on average across a whole bunch of people, thousands and thousands of people with chronological age. And then we can hopefully make some inference about people that don't fit nicely on that. So residuals. So we could think about people as a residual, right? They either are on that line or they're a little bit off. Some of them might be above the line. And so they might be aging a little bit more accelerated. And some of them might be below the line. They're aging decelerated. So they're aging less than their chronological age. This is work that Josh Danoff of Jessica Connolly's lab has been doing. He's been helping with the Cliff sample. He's been doing the work there as well. They took all the data that came from the Illumina chip. I'm not going to get into details with that. Josh has a lot of controls that he had to do. He had a lot of pre-processing of the data. This is a good advertisement to say that data don't just come off and they're magically turned into clocks, but there's a lot of quality control and there's a lot of work that goes into it to construct these types of clocks. And the clocks are published on, and there's tools to make them, and they're quite useful at this point. But what we can get now is we can get an epigenetic age estimator, which is a mathematical algorithm. And then it uses values assigned to the methylation state of specific CPG sites. And then it just gives you a single number. It gives you a number that shows where that person falls based off of where they should fall based off their chronological age. Steve Horvath of UCLA is the one that's probably most well known in terms of epigenetic clocks, at least the one that I encountered the most. He's on most papers of even newer clocks that come out. He's done a lot of work to show that these clocks are great because we can get them from various peripheral tissues. We can remove someone's blood and know that that relationship that we have with methylation and as assayed from the blood is the same as it is from other tissues, including things like brain tissue or including other tissues, lung, liver, et cetera. This is Horvath's multi-tissue age estimator. Really, I just popped this up here. I know you can't read all the different words here, but it's just different places across the body: liver, kidney, colon, brain, blood. And it's just showing, again, the chronological age and the methylation age based off of prediction from this, from a peripheral tissue in this case. And it shows this really nice correlation, which then gives us this confidence that even though we don't have access to perhaps our tissue of interest, if we're someone that studies liver, the liver, if we're someone that studies the brain, the brain. So we can still use this clock as a measure of epigenetic aging. Now these data are from our Cliff sample. These are data that Josh had generated. This is from Horvath's clock, and I'm going to talk briefly about that clock, but then I'm going to change gears and talk about a clock that's better for our purposes. This is just to show you that DNA methylation age is highly correlated with chronological age. So you can see some differences. So you can see the nice pattern of association, 60 to 80. And then you can see some variability in here. I'm going to show you this slide too, just because, as I said, Jess Connolly is involved with Joe Allen's group to also do this in a younger sample of people. And they were also run on this chip as well in this larger analysis. This is just to show you that group is, again, they're in their 30s, and you can see how they cluster here. And you can see the variability within the epigenetic age versus the chronological age. And then this is the VCAP sample here between 60 and 80. And you can see how they sort of cluster around there as well. One point I want to make that you might have noticed from the data that I showed you is that there's an underestimation of DNA methylation age in older adults. It's previously been reported. There's probably a number of reasons for this, but just as they have done this over time, they have found that you might underestimate age a little bit. So the way that you handle this is you don't just scrap it and say, okay, we have to start all over, is you just simply use age as a covariate. In this case, you control for age in your analysis, knowing that there could be a little bit of differences at each end of the spectrum when looking at these types of data. All right, since that time, as I said, there was a lot of money invested in making these types of clocks, and they've come really a long way. There's a number of clocks that have come up. DNA methylation age is Horvath's epigenetic clock. It's sort of the one that people think about, I think, most. It's the one that's like sort of the original. There's age acceleration residual, which is derived from the residuals of linear model predicting DNA age from chronological age. So instead of giving an age, you can just give a difference from chronological age if that makes your predictor better. There's age acceleration that you can derive from this measurement as well. So there's a number of ways you can go about this. I want to talk a little bit though about these clocks right here that I've highlighted. This is DNA grim age, which is a measure that was just recently developed within the last couple of years. And then there's this age acceleration grim, which is based off of this DNA grim age. What DNA grim age is it's an estimator of lifespan and health span. And it's essentially gives you this different way of thinking about it. So if I first built a clock, not me, but if I'm Horvath and I first built a clock based off of a bunch of people, and I'm simply interested in CPG sites that are highly correlated with chronological age, and I train a machine learning model to tell me which parts of the genome are important for that, I'm going to end up with a clock that's very good at getting close to chronological age. If I'm doing anything with regression, I'm trying to reduce residuals at all time, right? With DNA grim age, what they actually did was they took a bunch of different biomarkers of different plasma proteins that we know are important in aging processes, that they're important for health. And for each one of them, they calculated which CPG sites are basically variable as a function of these plasma proteins. And then they combined the various ones. So they took all the different plasma protein clocks that they made separately, independently, and then they add them together into this resulting elastic net Cox model, as it puts right here. So this is just a figure from their paper. Mathematically, we can't get into it. I wouldn't be the person to explain it to you anyway. But this is to say that they're taking a bunch of measures that we know report on things that are bad for you. So things that are in your system that are indicative of poorer health. And then they took a lot of their data from the Framingham Heart Study. So they were able to understand things like coronary heart disease and other causes of mortality. They were able to take different time points and understand when they got one time point versus when someone died, how long that was. So they were able to get time to death measurements. And then again, using this regression, they're then able to train up an algorithm to look at CPG sites that gives us this grim age, this time to death measurement, essentially. So it tells you that people that are lower in grim age, that they have this negative grim age acceleration, that they are doing good, that they are under the age sort of they are chronologically, they're away from that. And the people that are higher along there, this is a risk factor for many different health, all-cause mortality, time to death, time to cancer, time to coronary heart disease, et cetera. So this is a calculator that, rather than chronological age, is really sort of giving you information about an individual's health. Okay, so this is where I'm going to switch gears. We have a clock. We have these great data that were generated from Jessica Connolly's lab with Josh, as I said before. And now we're going to go back to the stuff that I do more of, which is functional magnetic resonance imaging, different fMRI scanning, and something that we've been interested in a lot recently, which is some of the properties of the functional connectome. In 2015, Emily Finn and colleagues wrote an important paper demonstrating that the functional connectome has a lot of individual variability in it that is unique to that person. So they called this functional connectome fingerprinting. And what they mean by this is that they can take a subject and they can run them on one scan, doing like an attention task. They can run them on another scan, doing like a resting task, and maybe doing an emotion task. And then they can take these runs. And so say you had 50 subjects in run one, and then you had the same 50 subjects in run two. You can train up a model based off of taking all of your first runs. And then that could then create a predictor so that you can see a scan now in your test database, run two, and how well can you predict who's who, just which subject is which. And what they found is that you can do this with remarkable accuracy, which means that the way that people are connected between different areas of their brain, when you look all across the brain and you take many, many connections, there's a uniqueness to this. There is a functional fingerprint, if you will. And also what they had found is that if you look at some of these networks, they're predictive of cognitive types of things that we might be interested in. For instance, the first thing they tested was on intelligence, scales of intelligence. Okay, so this is how they did this to begin with. I'll just show you that we did an analysis just like theirs to sort of wet our feet to see where we were with this. In the original analysis, they took data from the Human Connectome project, and they took resting state data, which is what we have as well. And they would take rest one, and they would use all the people, the 126 people from rest one, and then they would take the 126 people's rest two data and try to predict rest two from rest one. And they were 92.9% accuracy in identifying who's who, of course, in terms of the subject, which is, again, really great data in that case. Same way you can change what's the target and what's the database, so you can train up your model on rest two and then predict rest one. And again, it's similar with just a little bit of differences that you're going to get when you get into modeling. Our VCAP sample, one thing I glossed over at the very beginning, and I'm sorry for that, we collected structural data. We collected 2 scans of resting state data. So we collected rest one, and then we collected a functional scan, then we collected rest two. So they were about 8 minutes apart or so between these 2 resting state scans. And then we collected diffusion weighted MRI data at the end of the scan sequence. In this case, we had rest one and rest two. We found we had 94.5% accuracy and then 96.7% accuracy from rest two to rest one. So again, doing this functional connectivity fingerprinting on VCAP subjects, even though it's an elderly sample, they still show characteristics in their connectome that show unique variability that we can pick out that way. This is a kind of a messy plot at first when you look at it, this circle plot. This is just showing you a measure that the Yale group had used that we also employed, which is kind of useful. It basically asks, okay, so what connections are useful? Which connections are showing all the variability? So if we're training models and they're successful at predicting unique connectomes, there must be a lot of variability in all these connections. And some of them must be more variable than others, and those must be the ones that the model are using when trying to predict who's who. So the way that we do this is we take 268 nodes across the brain, and I'll show you a little bit more about these in terms of networks in a minute. But you take 268 little spots across the brain, essentially, we call them nodes, and then you create just a gigantic correlation matrix of the time series. So you have 268 by 268, and then you can draw all the significant connections to one another. In terms of these two measures, this differential power, this is showing which nodes are most variable and therefore most useful to the model. And then this measure of phi is showing which ones of these are just kind of common across all people, much less variability, strong connectivity in these networks, but not variable between individuals. So if you look at this one, you'll see, for instance, the motor cortex on the right side, right hemisphere and left hemisphere are pretty well laterally organized in this way. And there's a lot of connectivity between there, but it's not really variable between individuals. So it's just a property of connectivity of the brain in this case. Same thing you'll see here in the visual cortices as well. You get a lot of within network connectivity in this way that isn't that different when you look between individuals. So these are the connections that you would say are common across the group. Here, these are the connections that are most significant in terms of showing the most variability that is different amongst people, and that the models are really using when trying to differentiate this is person A versus this is person B. And here you see a lot of the connectivity coming out of this frontoparietal networks and down here into the medial frontal networks. And so you see sort of these thick strands, look like a big thick group of strings in this way. This is connectivity between those areas. And interestingly enough, when they did the test to figure out if this connectivity is showing anything, this is where a lot of the stuff was predictive of intelligence and some of these phenotypic differences that we think of as stable. So this is sort of a nice way of demonstrating sort of the power of the technique. All right, so using the connectome, if the connectome has some stability in terms of an individual difference measure, then we're in better shape than we are with traditional fMRI, with traditional bold imaging. So people complain all the time, and I see it all the time because I'm on Twitter and I read stuff about fMRI, but they'll say things like, you know, bold imaging, you know, any kind of regional activation is not reliable. They make lousy biomarkers. They're good for brain mapping, but not much else. And there's some truth to that, and there's some falseness to it. It depends on the way you think about it in terms of a psychologist. It's the behavior, it's the task, or the way you think about it in terms of a neuroscientist, it's not looking at the whole brain. So functional connectome and connectome predictive modeling tends to, instead of looking at these regions of specialization, and we know the brain has regions of specialization, and you talk to any neuropsychologist and they'll talk about this with us, but we also know that a lot of our complex behaviors are due to emergent properties of the brain itself, and we need to understand it from a whole brain perspective. So what we do with connectome predictive modeling is a unique sort of mathematical framework in which we take each subject's connectivity matrix. So we take that 268 by 268 connectivity matrix. We establish a strength between each one of those nodes. So we can establish a strength just by a correlation even. So you can just take an R value, do time series. And then we can take that, and then we can, for each subject, have a behavioral measure of interest. In our case, we're going to use age as our measure because that's going to be something that's variable. But you could use any individual difference measure in this case. And then so for this matrix, we're going to take an individual difference measure, correlate it with the strength of the connection, and then we're going to have this matrix that shows a correlation between strength of a node and the individual difference variable that we're interested in. And then we're going to do it as principled. We're going to use a cutoff where we are going to select only the most significantly correlated edges, the ones that show either the strongest positive or the strongest negative correlation. And then we're going to give it a score. We can sum all the edges. We could take the brain behavior relationship. We could create a regression equation, and then we can take data we haven't seen and predict a score from that data. And then we can see how well did our model do. So our model should be predicted versus real. in the case of predicted versus real, if we have a good model, a significant model, then our model that we made should have a really nice, tight sort of positive sort of correlation in terms of the way that we look at the model, the way that we verify the model in this case. Okay, so that's how we're going to go about doing this connectome predictive modeling. It's going to give us a chance to use machine learning to talk about where we can find individual variability in the brain and what connections are showing this individual variability. All right, I mentioned the Shen Atlas a number of times, so I'm just going to show it to you here so you know what I'm talking about in this case. When we talk about the Shen Atlas, I'm talking about a couple different things. First of all, I said 268 nodes. All right, so first of all, we're going to parcel the brain into 268 different parts. So this isn't honoring any kind of particular functional neuroanatomy. It's not honoring any kind of typical anatomical location. It's not the same as like when we do this with Brodmann's areas. Obviously, we're talking about different cellular layers. In this case, we're talking about just taking the brain and parceling it into a specific number of nodes. And there's any number of schemes out there that have attempted to do this. This is the Shen Atlas. This is a common one. They've also done what they call resting state networks. Resting state networks are data-driven networks. No one tells you what this network is. The data tell you what it is, essentially, and then they name it afterwards. What they mean by this is that we know that the brain has intrinsic functional connectivity. What I mean by that is I don't know anything per se, I don't have to know anything per se about true anatomical connections, but I can tell you certain parts of the brain have time series that are highly correlated with each other across time. So certain parts of the brain are integrated with each other across time. And this looks different than other networks that are sticking together across time. And what we can do is using something like independent components analysis or some other fancy way of taking this apart, we can identify various networks, areas of the brain that tend to stick together. And then they've done that in this case, they've created a data-driven functional networks, and then they've given them names. The names aren't always neat, because oftentimes they'll include various areas. They'll be called the visual one, but it might not necessarily be in the primary visual cortex, and that's it. The medial frontal will be in medial frontal areas, but you also see some temporal areas involved there as well. Again, the brain doesn't know how you want to parcelate it. It doesn't care. So in this case, it's going to have these functional networks that are talking to one another, and we're just trying to pick up on these properties, trying to apply a universal way that we can talk about it, so we can then talk about it across research groups and then make inferences about it. All right, so here's what we can do. So here's the first sort of point with this is where I'm going with this. The first sort of attempt that I want to talk about is let's use CPM to predict age during a functional scan. So this is just a proof of concept. I know from however long fMRI has been going on, since the 90s, I know that the brain changes with age. You don't need to convince me of it. I don't need to convince myself. There's thousands of paper about functional connectivity changes. Networks are different with age. Everything's different with age. So if I take two samples, an 18 to 30-year-old sample, which I happen to have because of our other work, and the VCAP sample, the 60 to 80 group, and I put them all into one model because they all did the same functional task. And then I ask, can we build a CPM based off of this functional task with these group of people where it can identify age, where we can find, basically we could do a reasonable job of predicting people's age with this, because that's how the model is defined. And the answer is going to be yes. I can tell you that because things change with age, and you're including so many ages that this is going to be great. So this is, Sam Brindley's been working on this as a part of another project, but this is just the results of this sort of analysis. What you find here is that we make a positive network, which is which connections predict higher age, and then a negative network, which connections predict lower age. And what you have here is that the overall model is highly significant, so you get this highly correlated sort of prediction equation. And then this is compared to, this is a random model. So we did permutation testing to test the significance of models. The positive model is highly significant. So the model that's looking for the positive connections, the connections that predict higher age is significant. And then the connections predicting lower age are significant. And this was a functional data run. This wasn't resting state. participants were doing a challenging social attention task while they were doing this. Just to show you what some of these look like, I took one network. This connectome, by the way, is a mess because there was so many connections because it had 20 to 80-year-old people. And as I said, they change a ton. So this connectome is just packed, right? So I took one network. One network is packed with connections in this case. This is the dorsal attention network, which is discussed a lot in the literature, very important for a variety of cognitive tasks. You can see slight differences here, obviously not going to be the same connections. The patterning looks somewhat the same in some ways, but it looks a little bit different in others. But essentially, what you're going to find is that the model is good at predicting both positive and negative, both old age and young age in this dorsal attention network, as you saw from that other data that I showed you before. The other thing we can do is we can look across functional networks. And here's where I want to make a stronger point. So anyone that knows anything about functional connectivity and aging knows some of the old dogma. That is, if I'm comparing samples that include people from 20 to 80, one of the things I'm going to tell everyone is that there's going to be more between network connectivity and less within network connectivity as you age. This is something that researchers have been saying for the last 20 years. They think about it as sort of a despecialization. So the idea from neuroscience was that you are more specialized when you're younger, that you have these networks that are, this network is face perception, face recognition, this is episodic memory formation, this is, attention, and that they're more distinct, more Swiss Army knife-like. And then as you get older, they just get kind of they move together like all the tendons in my body or something, just getting old, but instead it's the way that the brain is working, it's a property of the brain that they're becoming less specialized. And when we see this in fMRI, this is how this works out. Well, look here, this is exactly what you would predict then if that old adage is true. Is the yellow line here is each of these functional, so connections that are within a functional network. So this within network functional connectivity goes down with age. You can see that. So connections within network that I've drawn here with this yellow line, there's few of them in the older people, in the older prediction, in the positive network, the network that's predicting older age. Whereas the network predicting younger age, it's using tons of those connections within network, right? So you got all these 219, 175, 490 in the motor network. So this within network connectivity is really sort of an interesting sort of validation of that old adage in the literature in that case. This is a different way. This is called a Hinton plot of visualizing this. You could see both positive and negative edges. Again, negative edges are those connections that would predict lower age, and positive connections are those that would predict higher age. So the red and yellowish types of squares are going to be the connections between areas. So you can look at sort of between network connectivity here, and you can see some of these larger between network connections coming out. Again, this between network connectivity looks bigger as a function of age. This is something that's been reported on before, whereas this within network connectivity often looks much stronger in younger people in this case. So again, this sort of validates a lot of what we see in the literature. Now, what happens when we get rid of the 20 to 30-year-olds, the 18 to 30-year-olds? What if we just look in 60 to 80-year-olds? Using chronological age as an individual difference measure, CPM is unable to generate a significant model. So when you look at people just 60 to 80 in their resting state data, they're different, right? So just because their age is, all in there in 60 to 80, because there's so much difference between any two 62-year-olds, then we have a hard time finding anything meaningful. We have a hard time finding something meaningful in the brain that's going to drive connectivity differences. These models, this is the random model, this is the real model for the whole model. This is for the negative network, this is for the positive network. This is null data. That's what null data look like. That's what a failed classification model looks like in this case. So when we use CPM to predict chronological age in the VCAP sample, we can't do it. We get nothing. Now, when we use grim age, that epigenetic measure as the predictor, we get significant models. So the whole model in this case is significant. the negative model is significant and the positive model is significant. So what we did is, again, we took the 268 areas, we had connections between all of them. We took the strength of connections. So we only took the connections that were showing a connection to one another over 0.2. Then we correlated it with grim age acceleration, which we had sort of 0, you could think of as no acceleration. Negative scores were going to be younger, better living, let's say, better aging, and the positive scores were worse living. So the positive network was the people that are aging more biologically, and the negative network are people that are aging less biologically. So the R threshold was set at 2. This was run using 5-fold cross-validation. It's done 10,000 times in terms of the null model. So we do this a lot. We do a lot of iterations of this. There's a lot of principles in machine learning that we've been following to do this. It takes a long time. You got to do it on the Ravana server, and it generates these data for us. And here, what you have is, here's some circle plots again. I just want to show you some things that we're finding from here. These are demonstrating the significant edges in predicting these age, in this case, grim age in this case. The positive network, again, these are the edges that are associated with higher grim age. And this is the negative network. These are the networks that are in connections that are associated with lower grim age. Okay, so these are the various regions. You can see they're color coded. This is prefrontal, motor strip, but this is sort of how the key goes with this. This is just taking all that brain and putting it into a space that we can look at it. We can also overlay it on top of the brain. It's a little busy, it's hard to visualize. Now, let me just show you a couple more parts of this because I want to quickly make a point about it. One is that trending that we saw before when looking from 20 till 80 is not the same. We don't get this home run of, oh, it's just, you know, again, we find this within network connectivity is better in the people that are younger in terms of grim age. It's more complicated than that. What it really looks like when you look at this and you start looking at other aspects of the literature, there are some networks where this within network connectivity really is going to matter. For instance, the default mode network, one that's been written about in terms of the aging literature multiple times. The medial frontal network, the frontal parietal network. These are potential networks that this is important here, where some of these sensory networks are actually showing stronger connectivity within network in people that are aging poorly. And so when we kind of visualize this, we can then try to use one of these Hinton plots again, and we can try to make sense of some of these connections with each other in terms of the aging. And this is what we're going to do next, is to try to ask ourselves, okay, so these networks that we're finding with this super specific, really well validated and really great biomarker for predicting age and through connectivity and showing us differences in connectivity, which ones of these connections now might be explaining some of the cognitive decline metrics that we have in the VCAP sample? And that's where we're going to try to take some of these next steps with this. I want to show you just a few more slides of data, so I'm almost out of time here. But I want to show you another project that's ongoing. We're working with Ben Newman, who works with Jason Drewskill in the neuroscience. Ben's in the neuroscience graduate program. He does some work with white matter in DKI type of data and white matter hyperintensities. And what he's done is he's done a lot of white matter microstructure studies. And he's created this pipeline where he's been using some tools that are discussed in the literature, but what they do is they try to find a more fine-grained way of visualizing and thinking about white matter measures that we're getting from MRI and that we're getting from diffusion MRI specifically. And what you can do is you can get different metrics of the different types of tissue changes that are going on. So this is why it's a microstructure analysis. This is a white matter tractography. Sometimes you see these types of things from a VCAP participant. This is white matter fiber orientation distribution and white matter hyperintensities. So this is run on a VCAP participant. You see these little pockets here. These are called white matter hyperintensities. They have different volume changes. They have various ways that some of them are bigger, you know, and they're related to a lot of factors of aging. It's thought to reflect demyelination due to small vessel disease. It has some implications for cognitive decline, dementia, Alzheimer's, but it's also just a normal part of aging. You start to have more of these on your brain as you get older. So Ben has been taking the data that we have from the clock, from the Grim age, and what he's been able to do is he's been able to show that Grim age significantly predicts total white matter hyperintensity volume. So this can predict how much volume these white matter hyperintensities have when you control for chronological age, sex, and overall brain volume. So he's controlling for all the relevant factors in there. It's also significantly associated with microstructure. So I told you he has these different microstructures within the scan itself, within the voxels itself. We can take apart various contributions to these differences based off of different physiological processes. And it's been associated here with greater extracellular fluid infiltration into axonal areas, higher intracellular fluid, and lower axonal signal when controlling, again, for these other relevant details. So this is a low Grim age subject. Again, you see cohesive axonal signal, small overall white matter hyperintensity volume. So you can see this is a person that's aging well, Grim age-wise, and their brain looks to be aging well in terms of this type of metric. Whereas this is a person with a higher Grim age. You have these higher, bigger volume white matter hyperintensities, and you have these axonal areas are more infiltrated by this isotropic fluid. So there's more of this intracellular fluid. You can see more of the green on there. So again, this is matching up where you have these brain changes associated not with chronological age, but with this epigenetic Grim age. He's also done some ROI analyses, again, parcelate the brain into unique areas. In this case, there's 212 ROIs that he's used, run some general linear models to test whether or not these significant differences that you're finding in volume changes are significant when you can control for multiple comparisons, when you control for chronological age, and control for all these other things, as I said before. I'll just give you a quick peek at some of these data. Across the brain, you can find many different significant differences here. So significant extracellular fluid changes. These are areas where you can find sort of positive, so changes in positive direction, so associated with either more change or negative, less change in that case. This just shows you what this would look like. This is an ROI analysis of microstructural changes associated with Grim age. Again, you have extracellular fluid infiltration concentrated in the frontal lobes. There's some in the corpus callosum and left temporal lobe. So these are some of the regions of interest that are beginning to come out of this analysis. I'm not going to make any specific claims about these white matter data. These literally are like half a week old. Ben has just been looking at this. We've been all sort of working together on these projects, and so he's been sort of, we still have to validate several things going on, but it's promising, and it really tells a neat story about the epigenetic clock we're using and how this could give us some real specific information about the aging process itself. So the major take home that I want to put out there is that a lot of what we know with aging is because we studied 20 to 80-year-olds, 20 to 100-year-olds. This is both true cognitively and from a neuroscience perspective. One of the hardest things has been to study tighter cohorts, to study when we're actually interested in. When we say we're interested in aging, we're really interested in people that are older, right? So it's not enough just to say that this group of people, 60 to 80, look different than this group of people, 18 to 30. You do this in developmental psychology, incidentally, and people kill you, right? Like if I say a 20-year-old looks different than an 8-year-old, everyone says, I don't care. You know what I mean? 20-year-olds are different than 8-year-olds. Their brains are different. They must be different. So in this case, again, it's very helpful in terms of mapping and understanding brains changes over a long course of time. But with what we're interested in is diseases of the brain that are common within a particular cohort of individuals or a time period of individuals. We need to have more precise marks. And in this case, this Grim age mark seems to be uniquely qualified for the task. This is just, again, we've just begun to scratch the surface of these data, and it has us all sort of, like popping out of our seats a little bit, like, wow, that's cool. I didn't know that would work. It's really this very interesting sort of specific biomarker of age. And I think it's going to help us learn more about particular functional networks and maturation, and hopefully give us more insight into how these changes actually correlate with cognitive changes, which if you really look at the literature, we have some predictability, but we could do way better. So can we then use some of these networks and actually show some meaningful brain behavior correlations? And that's what we're going to be trying to do. Just wanted to thank again the research team, people that I mentioned today, obviously Jess and Josh, and Amalia with the functional connectivity, and Ben out of Jason's lab, and then also Morgan Lynch, who collected all the data, and Sam Brindley, who's doing some work as well with the older person data now as well. And then a couple of other people in Jess's lab who have done some other aspects of this project that I didn't talk about today, but it's a big team effort, and I want to thank everyone there. And the NIH paid for the scans, and NSF paid for some of the younger people's scans. And thanks again to the Brain Institute for allowing us to get the whole genome data, because it's been super fun for us to be able to see this. Thank you.
Jamie, that was really nice. I was fascinated by your white matter changes, because we've been discussing this, and if people who have hypertension, vascular risk factors, hypertension, diabetes, metabolic syndrome, tend to have those white matter changes, and more than normal aging, right? And I know that you guys, I don't know if you parsed that data out, but with the Virginia Alzheimer's Disease Center cohort, there may be opportunities to look not just at connectomics, but white matter changes, especially vulnerable populations who are more likely to suffer from dementia. We suspect that that's the hypothesis that Carol and I have been discussing, will likely have more subcortical white matter changes. So there may be some rich data coming.
That would be great, and I rely a lot on Jason and Ben. In that case, they do a lot of that work, and we're getting ready to have a conversation about some of the ways that we can capitalize on this more. But yeah, I think it looks super promising, and I know Ben has found a lot of interesting things. He has the BCAP data, and he plans to use some of it in his dissertation. So anything we could add to that would be amazing.
Jamie, this is Kevin. That was a fantastic presentation, amazing data. For calculating the GRIM age, what genetics data do you need? Like if you had whole genome sequencing data, would that allow you to do it?
No, Jess is shaking her head. She'd be the one that would tell you. What would you need in terms of?
You need the Illumina 850K epic array data, okay, or the 450K. The 450K works too, but there's a few sites that are different. It'll work the same, but.
Say that one more time. 850 or 450?
Or 450, yeah.
Okay, and then how is that different from? Sorry for asking such a basic question in the weeds, but how is that different from getting like, whole genome sequencing data?
Yeah, the epic arrays actually assay DNA methylation. And so if when you sequenced, you weren't looking for DNA methylation, that is, you didn't bisulfite convert to look to see what Cs were methylated, right?
Yeah.
So what you just have with whole genome sequencing data is the genetic data, which is super important as well, but you would need the epigenetic data in order to do this.
Okay, gotcha. And then if you had whole blood on people,
yep.
Okay, great.
But Kevin, relevant to what you're saying too, these clocks are being made for various tissues as well, and for a lot of different samples. Relative to kids samples, one of the things that Jess and I have been talking a lot about doing is there's a PED clock that uses buccal cells.
Okay.
Kids, and it's highly specific to changes that go on early in the lifespan rather than late in the lifespan. Yeah. So, you could build a clock on anything you want to, chronologically or some health variable. That's what's great is there's a lot of different ones built now. So if you want one on disease, then you go look for one on disease. If you want one that just matches up well with age, you can find one that matches up well with age. And so that's why some of the fruits of people's sort of labor of creating these things are starting to come together in a good way.
Gotcha. And then related to that, if you had the DNA cell lines from the Rutgers repository, could you do it on that?
Yeah.
Yeah, you could. Yeah, you just need to pull the DNA out of the cells, but yes.
Great. All
Yep.
right.
Yep.
Cool.
Yep.
I'm asking on, thinking ACE network stuff.
Yeah.
So very cool. Amazing. I hope you're planning on sending that last set of findings to a really, really good journal like, Nature or Science, because that's where I think they should be seen.
Yeah, we're really excited. I think some of it's coming out to be really cool.
Very, very nice work.
Hey, Jamie, you probably said it, and I just missed it, but to establish the connections between the different parts of the brain, are you using like a 3D bold sequence to image increased blood flow or changes in blood flow? Or...
No, we're just using regular, we're just using a functional sequence, just using, basically it's just functional MR, just using bold. And we're just looking at changes in bold over time in 268 different nodes from the brain.
And you do that under different tasks, or is this just resting state?
Resting,
FMR?
you can do it, it will work, the framework of it will work if you're doing a functional task. And so the original paper was showing some of the remarkable stability of individual differences across task type. But you can, depending on what you're interested in, you could do it with a functional task. You could do it with a task where like an attention task or something, but you could also just do it with resting. And most of the work that's been looking at these networks and aging have looked at it with resting state fMRI. So that's just people staring at a crosshair on the screen for 8 minutes.
Right,
But it's
cool.
capitalizing on these, as I said, these intrinsic networks that have been easy to identify in large groups of people. And, so the brain seems to have these sort of intrinsic functional networks that are not always anatomically, they don't always make anatomical sense either, by the way, but for some reason, functionally, they tend to glom together.
So, can you hear me?
Yeah.
Perfect. This is a really interesting talk. I'm very new to this field, so I don't fully understand what you showed. So the question I have is more theoretical. So you're showing that there's more inter-regional connectivity with aging, right?
Yeah.
So one thing that's lost from with aging is the plasticity, how plastic the brain is and how easy it is to learn, I guess, newer tasks. So do you think that this increased interconnectivity kind of just gunked up the system and makes it harder to form newer connections? And that's what leads to less plasticity in aging? Or do you think it's not a function of that at all?
Well, it's a good question. And there's different sort of ideas about this. And it's not settled by any stretch. So you kind of have a chicken and egg problem on some of it. Some people think that, with various levels of cognitive decline, because of maybe some efficiencies, even regionally in certain brain areas or brain systems, that cognition changes, the way you go about solving problems changes. I gave a grim picture of aging and cognitive decline over time. There are actually some things you get better at, by the way. Yeah. You get more, your verbal capacity is higher over time. There are certain sort of facts that you grow in, more, you have more information. So you actually change your problem solving a little bit. It's kind of like a speed accuracy trade-off. You trade a little bit of speed for a lot more accuracy. So some people think that some of these cognition changes are what's resulting in some of the functional connectivity changes over time as well, sort of, so small changes here and there are leading to different ways that we use our brain, and that's leading to a different reorganization. And then other people think that, yeah, maybe it's just that there's less efficiencies, maybe demyelination in certain areas is creating sort of harder connections across long areas of space versus shorter areas of space. But at the end of the day, it's also hard because we can't make great inferences about strength of connection. fMRI is not going to give us necessarily a strength of connection. These are most reliable connections for predicting the model. So it's basically saying that these node A and node B is really strong in terms of the variability there, really seems to be, capturing the strength seems to be trending with the older age rather than the younger age. But it's not like, it's not an absolute. It's not like something that has like a ratio level where it's like it's either all none or really high or really low. So we don't have that direct measurement in that case. And that's one of the hard parts with fMRI as well. But what we can do is we can think about this in terms of those systems, and then we can take the network and then we can start, we can basically computationally lesion it. You know what I mean? We could say, what happens if you remove these connections? Can we still explain performance? Or, you know, do we see that these connections were super important for an attentional task or something like that? So I appreciate your question, and it's an important one. And I wish I had a simple answer to it, but the problem is there's no simple answer to it. And there's like about 30 theories as to why that happens that way.
Yeah.
Roberto Cabeza, someone at Duke, has done things over the years and has talked a lot about cognition across the lifespan and different brain ring organization, if you're interested in some of
I'll
those.
look him up. Perfect. Thank you so much.
All right. Thank you, Jamie.